Hello everyone, welcome to Get My Class. So let's solve this question which says ELISA can detect option A antigens, option B antibody and antigen, option C glycoprotein, option D both B and C. Now let us understand the question. It says ELISA detects. So what does ELISA detect? So first, before we actually talk about what does ELISA detect, let us understand what is ELISA. What is ELISA? ELISA is nothing but enzyme linked. E for enzyme, L for linked, I for immunosorbent, okay. S for, like I and S together is immunosorbent, and A is for assay. When we say assay, actually we are trying to find out something, we are trying to detect something. So this is a diagnostic method. Diagnostic method. Okay. Now diagnostic method for what? It is a diagnostic method for the presence of, or you can say to detect any disease, which has the capacity, any disease where the you can say the infection is there, okay, any infectious disease, or even sometimes the disorders, like if there is any disorder because of which there is production of some protein which is not necessary, then that can be detected by ELISA. So the basic principle behind this is antigen and antibody interaction. We all know that. Okay. So the antigen, if antigen, we have to find out what will happen. We will add the antibody complementary to it, as well as this will be linked by an enzyme. Okay. So you know this is the antibody, right? Y-shaped antibody, we all know. Yes. And Y-shaped antibody will be linked with an enzyme. Suppose this is an enzyme. This enzyme can be this can be used to detect for the change of the color. If there is change of the color, that means this enzyme has been utilized there. And if this enzyme has been utilized there, that means this antibody to which this enzyme has been attached, that has been attached to the respective antigen or antibody to be detected. So if this is there, if this gives color, it will give change in the color. If this gives any change in the color, that means that particular antigen or antibody is present. Okay. So always remember this antibody, what we are talking about, this can be against an antigen or it can be against an antibody also. So you also get antibody. That means anti-antibody also. Okay. And now let us look at the options. Option A says antigens. Yes, it is used to detect antigens. Option B says antibody and antigen. Yes. Okay, so this makes more sense because here they have given both. And option C says glycoprotein. Glycoprotein, yes, because in some of the cases, as I told you, in certain disorders, there can be production of certain glycoproteins, or even sometimes the antigen, usually they are proteins, but sometimes they can be even glycoprotein. So what is glycoprotein? Glycoprotein will be the combination of both protein and saccharide, that is protein and carbohydrate. That is why it's called as glycoprotein. Okay. So basically, if you have to consider option D, that is both B and C. Why not A? Because already in whatever is present in A, it is also included in B. So you have to choose the option wisely. So the correct option will be D, because it consists of both antigen and antibody as well as glycoprotein. Okay, so ELISA can be utilized for detecting antigen and antibody, even certain other types of components like glycoprotein. So let us conclude the answer. The answer is option B. Okay, so I hope you understood the explanation. Thank you, everyone.